Hello and welcome, everyone. Thank you for being here today on defining healthy woodland soil. This is the first webinar of the series, or the Healthy Soil series. This series is made possible through the New Mexico Department of Agriculture Soil Health Program. And this webinar series is in collaboration between the Forest Stewards Guild, Glorieta Freedom Ranch, and Ecotone Landscape Planning. Before we get started, I'd like to introduce myself. My name is Gabe Kohler. I'm a program coordinator with the Forest Stewards Guild. Very happy to be here today and excited to introduce this content to everyone and our speaker, Jan Willem. Before we get started, I'd like to just kind of set some kind of housekeeping stuff for the webinar format here. If you could please keep your cameras off throughout the webinar. That will really help us kind of make sure the streaming is of the highest quality. If you would also please keep your microphone muted until the Q&A period. There'll be a question and answer period at the end, and I will tell you that it's all right to unmute yourself if you'd like to speak directly to our presenter. As questions might come up during the presentation, go ahead and just put those in the chat box. And you can see here on this diagram of Zoom logistics, there's a little icon at the bar. It's either at the bottom of your screen, or for some it might be at the top. But click on that chat button and it'll allow you to enter questions into the chat. And I will keep track of those and relay those to Jan Willem during the question and answer periods. We'll have a brief question and answer period halfway through the webinar for about 5 minutes. This can be a quick bathroom break, grab some water or whatever. And then we'll have a longer, more open format question and answer period towards the end. So yes, please park your questions in the chat. We're really happy to have questions. We'd love this to be an interactive webinar. And if there's clarifying questions, I can go ahead and answer those as we go. So if there's acronyms or things that you don't understand, I'm happy to just answer those on the fly in the chat box. I think that's all for kind of housekeeping stuff. As it says here, this webinar is being recorded and will be publicly distributed. The other two webinars, webinars 2 and 3 of this series, are on YouTube currently. And we'll give you the keywords to find those. So if you're interested in the content that we talk about today, please, we encourage you to check out those other videos in the series. Really great information in there. So without further ado, I'd like to introduce our speaker, Jan Willem Janssens with Ecotone Landscape Planning. Thank you so much, Jan Willem. And with that, you can go ahead and unmute yourself and we can begin.
Hello, everybody. If you cannot hear me, I will hear from Gabe, I suppose. It is wonderful. Thank you. I'm glad to do this webinar for those who haven't had a chance to hear it, and for those who are interested in hearing it again. I'm a landscape planner. I've been in New Mexico for about 27 years. I've been working in the fields of forestry, woodland restoration, and watershed and wetland restoration. And am particularly interested in the interactions between the different ecosystems horizontally across the landscape, and then vertically, how plants relate with each other and with the animals in between them, and also with the soil and the water below them. And because soil is so important for the growth of everything on our earth, it was a wonderful opportunity through the state Healthy Soil Program to dive deeper into this and explore soil aspects in relation to this particular topic of pinyon juniper woodland ecologies that has been my focus of study through projects and independent study over the last several years. And I'm very happy to have the opportunity to share that some of my knowledge and questions about these things with you today. And thanks again to the Forest Stewards Guild, the New Mexico Department of Agriculture Soil Health Program, and very much also to Brad and Kathy Holian of the Glorieta Freedom Ranch for making this possible. So the webinar will introduce the critical importance of soil in holding water and as the natural growing medium for plants and many other forms of life. I hope to explain the self-generating and self-healing capacity of the soil, and those capacities are particularly generated if the soil is not overly stressed. I will address how soil is a living ecosystem that we need to take care of with attention and dedication in order to allow it to come to its fullest potential of harvesting and storing water, supporting plants and other life, and providing many other services that we often take for granted. And it's my opinion that once we take care of soil, we will discover what healthy soil can provide for us and how it supports our individual and community livelihoods. In pinyon and juniper ecosystems, this means that soil health contributes to enhanced grass cover, tree health and vigor, and as a result, we may expect better wildlife and livestock forage, for instance, and pinyon nut crops, firewood supplies, and wildlife habitat. And we may also expect greater tree resilience during droughts and insect outbreaks. So those are some of the benefits of understanding the interactions between soil and the terrestrial ecology of these pinyon juniper woodlands. So this is part of a series. This particular webinar focuses on defining the health of woodland soil. And it's then followed by the ones that are now in the form of YouTube online about measuring woodland soil health and managing woodlands for soil health. What we're going to talk about today is, we begin with an overview of the Glorieta Freedom Ranch on Glorieta Mesa, southeast of Santa Fe. Then we're talking a little bit more about pinyon juniper woodlands, what they are, and how you can actually see different types of them. Then we're talking more about soil and what is soil and all the related questions about soil and soil processes. And then we're about halfway with possibilities for brief question and answer. Then I'm going into the 5 soil health principles as they are developed by the New Mexico Department of Agriculture, and how these 5 soil health principles can be applied through monitoring and land management. And that ends our presentation for today with questions and answers. So the Glorieta Freedom Ranch is a forest and woodland property located on the northern end of Glorieta Mesa in Santa Fe County, just on the border of Santa Fe County in San Miguel County. So here's the county line, and this is Santa Fe County, and Santa Fe is just outside the image here, about 25 miles east of Santa Fe. The property is part of the ranch at Powder Springs subdivision, and the area is located in the southwestern corner of the former Pecos Pueblo Grant. The property is surrounded on the north and east and south side by private property, which is white in the map, and on the west side by the Santa Fe National Forest, which is the green color. It comprises multiple parcels, and the property spreads across approximately 870 acres. Elevations range between approximately 8,000 feet on the flanks of the Cerro Las Escobas, which is kind of here where my pointer is, and then that's in the northeastern corner of the property, and 7,300 feet here on the southwestern, in the southwestern valley bottom. Brad and Kathy Holian are the owners of Glorieta Freedom Ranch, and the ranch has a history of homesteading, grazing, and woodcutting. In the past 15 years, the ranch has been managed for conservation purposes and underwent a series of ecological restoration and stewardship activities. The ranch operates under the banner of the Tree Farm program and offers to be a stewardship education and research site for regional land conservation groups, educational institutions, and the Pueblo of Jemez. And next I will play a brief video in which Brad Holian explains about the land. Okay, and then I need to mute myself. Let's see.
Hi, I'm Brad Holian. Welcome to Glorieta Freedom Ranch. We are right now on Glorieta Mesa, not far, maybe a quarter of a mile from the rim. The elevation here is 7,777 feet. My wife and I own approximately 900 acres in the Glorieta Freedom Ranch. We're surrounded by national forest and some private land, but it's mostly just us and the animals and the trees and the grass. The land has drainage off to the east of me here. It's called Padre Springs Creek, and it is an intermittent stream. Part of the historical beauty of this land is that not far from here, probably a tenth of a mile or so, is a hand-dug well from the 1870s. It's hand-dug down 35 feet through 2 4-foot layers of very hard sandstone. And it has water in it now. And we can possibly say that the water retention and the riparian restoration work done just upstream here at the Big Willow riparian area has helped to bring that water down the Padre Springs drainage to the well. Our goal, of course, is in all of this is to, with the forest thinning, with the introduction of cattle to graze on rare occasions, all of this work produces a sponge in the grassland area that absorbs water and goes into the aquifer that's feeding that hand-dug well. Even further beyond that, about a mile from where I'm standing, is the original Padre Springs, and it's known locally as Pozo Hondo, the deep pool, and it's probably 20-30 feet deep and 50 feet long in a deep canyon of that same hard bedrock. And so we're actually feeding that water down through Padre Springs Creek. It's been absorbed here in the soil, healthy soil, and goes into the aquifer, and it doesn't just come running off quickly and gets.
Jan Willem.
All right. Yes.
Oh, it just ended abruptly. I just wanted to make sure, but it seems like we're moving forward. Thank you.
Yeah, no. So this is right. It ended in a strange way, but it was the end. Okay, so looking at pinyon juniper woodlands on the property, uh, it's important to realize that woodland ecologies are very diverse, and their diversity is directly related to the diversity of soils and the need that we need to identify different soil types. So pinyon juniper woodlands come in many different types of ecological communities, and these ecological differences are strongly related to the soil types, different water regimes, underlying geology, and climate. And climate, again, expresses itself in elevation and solar aspect. And for instance, on the Glorieta Freedom Ranch, we find persistent pinyon juniper woodlands, and I will explain later what they are, what that means, persistent woodlands. We find alluvial fans with grass and forbs. We find wetlands and riparian areas amidst the pinyon juniper, valley bottoms with grassland and scattered pinyons and junipers, and Ponderosa pine stands with a pinyon juniper understory. Um, Vegetation types and vegetation health can be an indicator of soil health and vice versa. Soil health influences the vegetation health. So that's why distinguishing different vegetation communities and soil types is critical to understanding their health. And a difference in vegetation community must always be observed in relation to its soil conditions to consider whether the vegetation and soil are supporting each other or not. So that's what we're going to look at next. So first, a little bit more about pinyon juniper woodland ecologies. They're one of the most important and expansive forest types in the western United States, stretching from western Texas in the southeastern corner of the country or of its area across the country, all the way to central Oregon, across about 10 states, as indicated in this image here. Pinyon juniper woodlands are very diverse, and to better understand pinyon juniper woodlands, it's useful to identify different types of woodland ecologies. As I mentioned before, there are persistent woodlands, which are found where local soils, climate, and disturbance regimes are favorable for pinyon juniper or a mix of both. The soils are typically coarse in texture, and these persistent woodlands are typically dependent on winter precipitation. Fire regimes have always been infrequent in these woodlands, and it must be said that for New Mexico, particularly, the New Mexico Forest and Watershed Restoration Institute also identifies open woodlands, because here in the Southwest, or in actually the southeastern range of the pinyon juniper ecologies, we have a more dry type of pinyon juniper ecologies that spread out over, that spread the trees out in lower densities across the landscape. The second type is the pinyon juniper shrubland, or wooded shrublands, and they're found where local soils and climate support shrub communities, but trees can increase during moist climatic conditions and periods without disturbance, such as fire, and then decrease again during droughts or following disturbances. Soils can have a variety of textures, from fine to coarse, and again, also here, winter precipitation is typically the most important in this ecosystem type. Then there are pinyon juniper savannas, where the local soils and climate are more suitable for trees and grasses. And these ecosystems occur on finer textured soils and in areas dominated by summer precipitation mostly. And it's logical that low severity fires may have maintained low tree densities because disruption of fire can be ignited through the grasses and then run into the trees. And this was mostly so probably before the disruption of the fire regimes following the Euro-American settlement. But information about this is insufficient to support any confident statements about historical disturbance regimes in these savannas. So furthermore, it's important to identify that here in New Mexico particularly, we have variations We also have open grasslands with some pinon and juniper. Some people say encroached, but some believe that they were naturally there. Then there are typically juniper savannas where you don't find any pinons. They're typical for central and central eastern New Mexico. And then, like I mentioned, Ponderosa pines with pinon juniper woodland beneath or alternating with each other. Pinon juniper ecologies were traditionally also agricultural in use. They were used for firewood, livestock forage and shelter, for harvesting pinon nuts, for harvesting fence posts and other utilitarian wood materials, and for hunting, for small game and especially deer hunting. So, and again, it's a very large ecosystem with many different types, and it's just in the last 10 to 20 years that we've begun to distinguish between these types so that we are better able to manage them and also start discovering how they are related to soil and other environmental characteristics. Here are the examples with persistent woodland here in Santa Fe County on Ortiz Mountain at 8,500 feet. It's very rocky, steep terrain, medium dense canopy cover, and very patchy structure. Then towards the north, it occurs at lower elevation at 7,000 feet. For instance, at the Mesa National Park in Montezuma County, just across the border in Colorado. Also, this terrain is very rocky, somewhat with loamy depressions, rolling terrain, and a very dense woodland structure. Fire regimes there are measured to be between 400 and 800 years. Then the pinon juniper shrubland. Here an example of Wind Mountain at the northern end of the Rio Grande del Norte National Monument, northeast of Tres Piedras. At 8,300 feet, definitely winter precipitation induced, and the terrain is loamy, nearly flat, and has a lot of sagebrush as the shrub component. Then the Mesa de la Cejita, northeast and north of Truchas in Rio Arriba County at 7,300 feet, where there is a different shrub component that includes mountain mahogany, Apache plume, cliff rose, Fentler bush, etc., among the pinons and junipers. And then the pinon juniper savanna. Like here at Juniper Hill Center, east of Las Vegas in San Miguel County at 7,000 feet on a limestone plateau, very gravelly, clay loam soils, rolling terrain. And this pinon juniper savanna eventually turns into a pure juniper savanna if you move more to the east, for instance, around Conscious Lake. Then also in southern Santa Fe County, we have good examples of pinon juniper savanna south of the Ortiz Mountain, but also north of it in the Galisteo Basin at around 6,500 to 6,700 feet. It's gently sloping, stony to gravelly, and has a variable density of pinons and junipers depending on drought and land use. And then finally, we'll find on the Glorieta Freedom Ranch and also throughout Santa Fe County the pinon juniper woodland with a Ponderosa pine overstory. And if you have taken hikes east of Santa Fe, you find a lot of that in the Ponderosa Ridge area, for instance, or the Wilderness Gate area. It's typically pretty steep and rocky, medium dense, Apache structure, and Ponderosa pine overstory that shows that the trees are pretty old. So the fire regime has not really gotten to both the Ponderosas and the pinons. So you both find you find trees of 300-400 years old in these areas. So that leads us then to also look at the soils under these different pinon juniper types. The soils on the Glorieta Freedom Ranch include loamier soils on grasslands, in the valley bottoms, that is soil type 702 and Web Soil Survey, and that supports mostly grass forbs, some Rocky Mountain juniper, not the one-seed juniper, but the more loose, not prickly Rocky Mountain juniper, some Ponderosa pine, and occasionally some piñons. Then soil type 703 is a variation of deep, loamy and fine sandy soil with rocky outcrops in a rocky underground, which you find in arroyos and alluvial fans, also with Ponderosa pines, Rocky Mountain juniper, and much more piñon and patches of grass in between. Then on the mesa slopes and shoulders, we have loam and clay loam soils with a petrocalcic soil component that is basically caliche at depths of as shallow as a foot to 5 feet deep. And here we find piñon, one seed juniper, not the Rocky Mountain juniper, but a one seed juniper, and oak. The water holding capacity is pretty low here, and a lot of water runs off, and the trees don't have good access to groundwater, so you get a much more spindly woodland forest. Other soil types are, and those are the more predominant ones of Glorieta Freedom Ranch, soil type 707 with many different varieties, which is more sandy on the lower slopes, with a lot of piñon, juniper, oak, and grass in a patchy, groupy structure. And then in the southern and western part of the range, on foot slopes, you have soil type 708, which is more loamy and clay, with piñon, juniper, oak, some grass, and not as much Ponderosa pine. And then the plateau shoulders, cobbly loam and fine sand in the northeastern part. They do have a lot of Ponderosa pine, are more north facing, and have some grass, and here and there some piñons as undergrowth. So you see how different locations and different aspects lead to, and different soil types then lead to different vegetation types and a different structure of openness or closeness, you could say, of the woodland structure. Understanding those general ecologies and relationships between the vegetation and the soil, let's now look more closely at what soil really is. Soil is defined in many different ways, but here is one that I like to use for the purpose of this webinar. Soil is a natural body comprised of solids, including minerals and organic matter, liquid and gases, that occurs on the land surface, occupies space, and is characterized by one or both of the following. First, horizons or layers that are distinguishable from the initial material as a result of additions, losses, transfers, and transformations of energy and matter. And second, or the ability to support rooted plants in a natural environment. So basically, the most important characterizations are this layering and the fact that soil supports plants. And that makes it really different from dirt and just the mineral parts of soil. And we'll look at that a little later. So soil is formed from the interactions between 4 different spheres on Earth. It depends on the balance and flow of the components and the processes from all these 4 spheres. That's the hydrosphere, of course, adding water, the atmosphere, offering the gases and temperature, the biosphere, offering everything that is living organisms, and the lithosphere, which is the mineral part of the Earth. It's also influenced by outside forces, time, particularly because we are looking at soil not as just one condition, but a process over time. Topography, and particularly that the gravity that pulls things down, both water and soil particles, and that topography leads to sedimentary soils, for instance, stacking on top of each other and forming soil, and people. So the influence of people has been always very important in forming soils. That's how we have agricultural soils. So all these interactions form that pedosphere, or the soil environment, in the middle where all these spheres overlap. So, and again, this is just not only a condition soil, it is a process of continuing interactions based on the processes and exchanges that happen in each sphere. It's a very dynamic way of thinking about soil. And that leads us then also to distinguish how soil is different from rock or dirt. Soil is a complete ecosystem. It's alive. And with the organisms that are in the soil, the organisms are mutualistic, symbiotic, or competitive. So they help each other, they live together, or they compete with each other. And in the mineral matrix of the upper part of the earth, that's only where the soil is. It's really in that very top couple of feet, really, of the Earth's surface, whereas dirt can be existing deeper down, and rock for sure. So that's how it is significantly different from rock and dirt. And it has proportional amounts of living biomass versus no life at all, or organic versus mineral material. And these differences in these proportional amounts define the different soil layers or soil horizons that are essential for identifying soil. And you can also identify them based, these soil horizons, based on different colors. And we're going to look at that next. And I'm trying to move forward. Okay, there we go. This again is a video. Well, it doesn't do it. I need to go back.
Here on Glorieta Freedom Ranch, we're looking at soils in Pinon juniper woodland. In some locations, the soils become exposed as a result of little gullies that have formed in what probably were cattle trails. This gives us the opportunity to look at a little soil profile in a soil pit here at the edge under some native grass. The soil cover consists of plant litter, decomposed plant litter, which is all the black organic matter that you see in between the grasses and the sticks. And then we're going to zoom into this soil pit here, where you see the little rootlets of the grass at the top with very fine threads that maybe move slightly in the wind, which are, as I understand it, mycelium threads, mycorrhizal fungi associated with the rootlets that hold soil together. Now we're getting into the rooting zone of this soil, and the roots go pretty deep until the bottom of the spit, which is about a foot deep, where we pretty much reach the mineral soil. And you see maybe slightly the color difference, where the bottom of the pit is a little bit more reddish. And the top has this grayish layer, gray blackish layer of organic matter that's associated with the decomposition of roots.
So looking at this in a little bit more schematic way, to summarize the video, it is important to identify in soils these different layers. And typically we identify the O horizon, which is the organic zone, consisting of plant litter or duff or any broken down plant parts that are at the top of the soil. Then immediately below it is the A horizon, which is the first important part of the soil, or basically the topsoil. And that consists of particulate organic matter, very fine organic components that are beginning to break down. And in this area, this upper soil or topsoil area, we have very high biological activity. Pretty much most of the microbiome happens in the first couple of inches of the soil. Then below that, the color changes radically from this gray-brown or blackish part to the mineral color of the soil, but typically with a little bit more brown or red in it. And that is because of more organic matter that happens as associated with the roots. And this area is called the B horizon. It has more limited particulate organic matter and has some mineralized carbon associated with the roots in these root channels that are going down. These roots are going down typically to reach water, but also to extend and with the water and the fungal activity, bring mineral ions up that fungi break down into the soil and that are essential micronutrients for the plants. Then lower down, we get into the parent material, the gravel, the sand, if it's sedimentary or fractured rock material. And there might be some roots in there, and that's then the C horizon or the C1 with rooted parent material. And then below that, a C2 horizon where there is basically no life. And below it is the R horizon, the parent rock, that is not affected by the soil at all, that is not alive, and that where basically we don't say it's part of the soil. So this lower soil in the middle has biological activity, but tends to be more geological and hydrological in its characteristics, remembering of those 4 circles that I showed. And then lower down, the C2 and R horizons have no biological activity and are only parent rock material, either solid in R horizon or somewhat fractured in the C2 horizon. And therefore, they're actually not part of the definition of soil. So in the 20th century, especially the first half of the 20th century, there was a debate, maybe mostly in Europe, but maybe also in this country, to what extent soil science was part of geology or biology. Well, in my upbringing in the Netherlands, it was often seen as part of geology, but that has really changed in the 20th century. In a book of tropical soils, a Dutch professor wrote together with his, I think, British counterpart, in the beginning was the rock, and the rock was the mother of the soils. Well, it's an interesting statement. Nowadays, I think few people will see it that way. And soil science is really a multidisciplinary science between all the different spheres with their science fields that I explained earlier. So the approach of looking at soil as a outflow of geology is still relevant maybe in understanding processes of mineral soil formation, which occur between the R and C horizon, the breakdown of mineral soils and providing ions and micronutrients to plants, and also in the context of soil erosion and sedimentation. But we have definitely come to understand soil science very much as an outflow or as a biological activity, and therefore also related to agriculture, because soil is definitely also a growing medium for land plants and cultivated plants. And in the last century, but especially the last several decades, we have discovered how important soil is in relation to all the life it contains. And therefore, soil in the last few decades has been approached from an ecological perspective as an ecosystem and a microbiome of countless microorganisms, including bacteria, fungi, protozoa, nematodes, worms, insects. And there are billions of soil microbes in a teaspoon. So we really are looking at something that is very much alive and that is at a microscopic level, an ecosystem in itself. We know now that the interactions between these ways of understanding soil explains very much about the processes of soil formation, soil building, and that the exchanges of minerals, water, and energy that take place in the soil in relation to plant life and atmospheric life are really mediated by the soil biology, the soil microbiome, and among them, especially the fungi. So, but just to take a snapshot in time to understand what soil is made up of, it is important to still understand that it is made up in principle of four important key components. That's the inorganic mineral matter, which is about 45% of the soil volume, and can sometimes be a little more, depending on how much organic matter we have. Organic matter is typically about 5% and includes all the microorganisms and macroorganisms living in the soil and their breakdown products. And in poorer soils, like here in the desert ecology, the percentage of organic matter and life is a little less than 5%. In other parts of the world that are very rich, it can be a little more. And then the other 50% are made up of water and air, and they are variable percentages, of course. When it's very dry, you have far less than 25% of water. If it has rained or after snow infiltration, there is more of that, and that presses the air out so that then the air component becomes less than the 25%. But roughly together, they make up 50-50, each 25% of the total soil components. Now, soil can hold water because of openings between all those soil particles. And there are basically three sizes of soil particles that we identify. Those are sand particles that are between 2 millimeters in size and 0.05 millimeters in size. Then silt particles, which are from 0.05 millimeters to 0.002 millimeters. And then clay particles, which are very fine. You can barely see them. You have to really use a microscope. They're smaller than 0.002 millimeters. And so that then leads to a mixture of soil particles that we call the soil texture. And depending on how the soil is being, how these particles are being structured, you get these openings, the pores that are between the soil particles. So large particles, such as gravel and sand, have big pores. And small particles, such as silt and clay, have small pores. And when water drains away from the pores, some water clings to the particles because of the capillary forces, which are the physical adherence of water to objects, and osmotic forces, which are the chemical adherence of water to molecules and ions. And together, these forces are called the matrix suction, or the suction force of the mineral soil matrix. And the condition in which all water has, when all water has evaporated or drained away, especially in I think the official definition is when it has drained away due to gravity, while the matrix suction is retained to the maximum amount, is called the field capacity. So that definition of field capacity is, imagine a soil that was filled with rainwater. Then that water drains away, and then still some water adheres to these soil particles, maybe expressed in these dark blue lines around the sand grains, and the lighter blue between the clay particles in this image to the left. Once it is in that state, it's called that the soil is at field capacity. Plants can still suck water from the surface of those mineral particles up to a certain level when the matrix suction is stronger than the plant's suction to suck up that water. And this moment of the soil's water availability is called the wilting point, because when transpiration in the leaves removes more water from the plant, the plant will begin to wilt because it cannot replace that water in the plant from sucking up more water between the particles. Now, difference between the field capacity when a maximum amount of water hangs on those particles and the wilting point, which is the point where the plant can no longer suck it up, that difference is the water availability for plants in the soil. And much more water is retained in fine textured soils, and plants can typically absorb, let's see, I'm not explaining this well. Much more water is retained in fine textured soils, and plants can typically absorb much of that water. However, the matrix suction in fine textured soils is much higher, and therefore the wilting point is reached earlier. And that is when there's still much more water left in the soil. So clay particles basically hold, well, actually, ideally, it is loam, silt loam, that holds actually the highest amounts of water, and where plants can extract the highest amount of water from the soil. That has to, basically, the soil holds the highest water availability for plants. Sand grains are so coarse that not much water adheres to it, and plants can actually, and once the wilting point is reached, there is not much water to be sucked from the grains at all. Clay particles also hold a serious amount of water, but they hold that water so strongly that at some point the plants cannot suck it from clay particles. So it is really important to understand the texture to understand the water availability for plants. So then the question is, how can we improve soil texture and soil structure? Soil structure basically being the clots that are formed and the pores and openings between the soil clots that are in the soil. Well, of course, you might think just add water, but no, not yet. It is most important to add organic matter and, of course, life forms. We need to add those elements of the soil microbiome. Well, why is that? Because organic matter and life forms help create a diversity of pore sizes and directions. It creates that structure. It basically creates a greater diversity of openings, opening sizes, and connections between pores in the soil in a three-dimensional format. And it mixes the organic matter throughout the soil horizons, much deeper than it normally would stay in the topsoil. And optimal root penetration is achieved then also at greater depth, so that if you have more openings in the mineral soil and there is more organic matter, then roots can get in there to grow and take part in the exchanges with the microbiome that they in fact feed. And you get then also more life forms and biological activity, and that then starts a process of humification, of breakdown of the organic matter, the breakdown products of all the life forms in the soil that eventually form a stable organic component called humus. And we also get increased ion exudates from fungi and other microbes and from the biochemical soil activity. That is really important because these ion exudates eventually form the micronutrients for plants. And then when all those components are in place, you have greater water retention due to the diverse structure of that soil, adherence of water to organic matter, and the connections between pores and multiple ions that also help with this water adherence in the soil. as I mentioned, that ions are one part of the matrix suction besides capillary action. I want to touch very briefly on soil acidity because it has something to do with the organic matter. So good amounts of organic matter and active humidification processes lead to a neutral soil of about 6.5 to 7. And if there is too much or poorly decomposed organic matter, the soil tends to get a little bit more acidic, which is a poor environment for insects and metazoic fauna, and that slows down the humidification. You will still have fungi and bacteria to do the job, but not the other components that we also need to break down the coarser parts of the organic matter. If there is too little organic matter, you will see that the soil pH might go up and it will turn more alkaline. And if it exceeds 8 in pH, then it becomes a pretty poor environment for soil macroflora and macrofauna, and you get pretty much a strong decline of biological activity, and plant life becomes then greatly impeded. We'll do a soil test exercise later in the webinar. So what do we know about pinyon juniper soils and woodlands, soils in pinyon juniper woodland soils, ecosystems? Well, there is still much to be discovered about soil conditions and soil health in pinyon juniper ecosystems. We know that soils in pinyon juniper develop in direct connection with tree species. We know that the trees contribute organic matter on the forest floor and deeper down. We know that there are, in the rooting areas, root-associated microbiota, or RAM, and root-associated fungi particularly, that are specific for each tree type and shrub species. For instance, mycorrhizal fungi for pinyons and arbuscular mycorrhizal fungi for junipers and other shrubs. Tree densities lead to specific soil moisture conditions and evapotranspiration rates, and also to different soil enzyme activities. So tree density changes through thinning impact what's happening in terms of the microbiome, the water components through evapotranspiration, and the soil biological activities underground. Healthy woodlands with healthy soil are called resource conserving communities and have ample vegetation and ground cover to limit runoff and soil loss and promoting the eco-hydrological resilience. But if that is being impacted, then you get that the entire ecosystem above ground and underground is going to decline, and that has mutual relationships between each other above and below ground. Healthy pinyon juniper ecosystems accumulate carbon in trees, in the litter, and deeper down in the soil through soil organic matter, and in microbes. And mineral carbon sequestration occurs through plant growth and soil microbial activity, and even in biocrust, cryptogamic crust, which are symbioses between various combinations of mosses, cyanobacteria, lichens, algae, and microfungi. And they also sequester carbon. And there is much of that in pinyon juniper landscapes, and they should not be underestimated. And the types of carbon in the soil and sequestration rates depends on tree densities and on the amount and sizes of openings between the trees. Old growth trees, especially juniper, are the largest source of carbon sequestration in pinyon juniper woodlands. And pinyon juniper woodlands may be the most important sink of carbon in southwestern ecosystems in our time of climate change, because they cover so much terrain, more than 50% of our forest cover, and they are not burning as much as Ponderosa pine forests. So, and because they have this very diverse ecosystem with shrubs and grasses and various layers of trees, that gives a very rich soil microbiome. The opportunities for carbon sequestration in a healthy woodland system are therefore pretty high. So we're now a little over halfway. If there are any questions, I am able to entertain them right now. So are there any questions at this point?
I'm not seeing any come through the chat. If anyone has a question, please enter it into the chat box now, and I will relay that question to Jan Willem. If not, this is a good time to take a little break, stretch your legs. But yeah, we're happy to take any questions.
Chat box is not working.
Oh, it's not? Well, go ahead and I guess just go ahead and voice your question now then, if it's not working for you.
Okay. Are these visuals available? Because a lot of information. I'd like to be able to get a hold of a lot of these slides.
Yeah, if you email me, and there will be an email address at the end, I can email you a PDF. And we're going to also make this available as a YouTube movie online.
Mm-hmm. And the other two webinars will be are already on YouTube as well, which are, we can give you the keywords to search for towards the end. We have a question here coming in the chat from Robert Templeton, who would like to know the elevation of the ranch in Pecos, Freedom Ranch.
The ranch is around 7,700 feet high, but varies. At the lowest end, it's 7,300. At the top end, close to 8,000 feet. And most of the slopes are having a southern or western aspect, but some of the slopes of the mesas on the eastern and southern side have north-facing slopes as well, like actually one in this picture. And it gives a much denser and diverse vegetation cover. Any other questions?
Last chance here before we move back into the presentation. Okay. Yeah, it looks like that is it for these questions at this point, Jan Willem.
Great. So I want to bring back this image where we're talking about soil health, and we're going to go to the 5 soil health principles. And what is really important here to understand again that soil health is not necessarily only the presence of the 4 components, water, gases, temperature, living organisms, and mineral soil. It is more importantly the balance between those components that leads to a health in the soil. If we take one of them away or diminish their influence, the health goes pretty much down. I mean, you can imagine that a soil that doesn't get enough water doesn't have enough life to support the microbiome in the soil, but also the plants that sink their roots in that soil and do the activity with the microbiome to have all these interactions that support both the plants and the entire soil micro ecosystem. If, for instance, the minerals, the rock component, are very hard to weather by the fungi, then you get a pretty poor soil because it doesn't hold any water, you don't get any growth and micronutrients, and so on. So you see how the balance between all those different elements is really essential. And therefore, we need to really start looking at soil as a process, as almost like a little microbial factory where a lot of things are happening, and that these processes also have to be balanced and have to be actually in a pretty speedy process, so that the outputs of these processes can be taken up by the plants, and that breakdown products are not lingering but taken up by microbes to be converted again, and so on. So think about soil as a process in time, with this fourth dimension of time is a very important component. And therefore, we're looking at soil health principles as the balance between the spheres and their products, and the processes that are happening, that each of the spheres are contributing. So the biosphere producing plant and animal litter products and the decomposition processes, the hydrosphere offering water and having water storage and water absorption in the soil, the atmosphere offering gases and heat with gas and heat exchanges in the soil, and the lithosphere offering minerals through the mineral weathering. And the pedosphere, the soil itself where everything happens, forms then stable or soil organic matter in a biochemical conversion process. And that leads to many benefits for everything that lives. It offers healthy processes for the gestation of life, basically the germination of plants, the growth of plants and animals and microorganisms, reproductive cycles of all organisms, including humans, the vitality of all living matter, and then of course the decay of it all and the cycling of it. And so the benefits are at the level of ecosystem flows, the health of all organisms, and even the level of molecular flows, leading then to plant health, animal health, and human health, but also to water storage and exchanges of water between different life forms, chemical exchanges, basically if there are pollutants, the microbes will actually break down the pollutants or isolate them. There is a lot of buffering happening, buffering of microclimate, buffering of water, buffering of pollutants again. And so that is, it's a regulating function on our Earth where we all live. Well, then you may ask, how can we lose that soil health, right? How do soils degrade in health? Well, we've seen atmospheric changes over the last 50 years, particularly climate chaos, where we have increasing atmospheric extremes in the form of enormous amounts of water coming down or not enough at a time, long periods of drought, increases of temperature, and so on. Out of season cold spells, and soil moisture imbalances. That variability in the atmosphere leads to changes in the soil. The structure gets more compacted. Parts of the soil dry out and cannot absorb as much. Part of the soil erode, leading then to a decay of the soil typically is. And intensive human uses, cattle trailing, tire tracks lead to compaction, for instance, and to erosion that also affect the soil health. Poor plant growth is a result of that, and if plants don't add their part to that soil activity, then we get a further decay of the health of the soil, especially because the decay of the root activity of those plants. And eventually, when soil cannot hold itself together anymore, the structure and texture falls apart, a little bit of water, hoof action, wind will help carry soil off, pulled onto it also by gravity, and that is erosion. and then we lose soil all the way to the bare bones of the landscape, which is the rock material. So then the question, of course, is how can we keep our soils healthy? Well, a healthy soil is defined as that which enhances its continuing capacity to function as a biological system, where it increases its organic matter and improves its structure and water and nutrient holding capacity. And we've discussed those components. And so that has led the state to formulate 5 soil health principles. And by implementing each principle, and even better, keeping all those 5 principles together, it helps keep the soil healthy in any given environment. So applying those 5 soil health principles guarantees that the soil maintains optimal structure, remains resilient, meaning self-healing after stressors, and sustains its biological functions. So it's important, first of all, to keep the soil covered. That's the first of the 5 soil health principles. And here in this image, you can see how that could be done by having plants grow or by spreading mulch in the form of, for instance, chips that are covered, that are a result of forest thinning. Soil cover relates to the presence of living plants and of organic and mineral mulches, such as plant litter, woody debris, and stone material. And how do you think these cover materials keep the soil healthy? Well, think of a protective skin. Think of the exchange of cycling of materials. Think of their role in water retention, and think of the growing medium or habitat for plants and animals and their interaction with the soil that's being covered and protected this way. The second one is minimizing soil disturbance and external inputs, and especially on farms and in fields, this is of course important, but also in woodlands. It relates to activities that impact soils above ground, such as turning the soil, compaction by hooves and wheels, removal or deposition of soil, and cover or spillage of trash, oil, chemicals, or construction materials that inhibit the natural soil functions. And minimal external inputs expand on that last aspect of minimizing disturbance in relation to the addition of chemicals, particularly fertilizers, fungicides, herbicides, pesticides, to the soil to change the selected soil processes. So of course, the disturbance and external inputs can change those processes that we discussed and then help or induce the degradation of these balances that are an expression of the soil health. Well, how do you think the minimization of such disturbances and external inputs keeps the soil healthy? Well, think of the natural biological processes that form the structure of soil and direct soil ecological functions and support many organisms in the soil. Think about the natural symbiotic processes in the soil, for instance. Then the third one is maximizing biodiversity. This relates to the multitude of diverse life forms above ground and underground that interact with and depend on soil for their survival, rejuvenation, and resilience in their interdependent relationships. This includes all life forms, from fungi to plants to animals, including their genetic richness. And how do you think maximum biodiversity keeps the soil healthy? Well, think of the many life forms in the soil and how they play a role in the digestion and cycling of products from life forms above ground, and how some above ground life forms have life cycles in the soil or depend on soil organisms for food. The fourth principle is maintaining a living root, and that's in response to having crops that really don't develop much rooting or that are harvested after a couple of months. So living roots relate to perennial plants that photosynthesize above ground and contribute to the carbon exchanges in the soil. Think about how living roots of perennial plants play a role in water and mineral exchanges with soil organisms, or how the symbiosis of soil organisms in the cycles and webs of life between atmosphere, water flows, mineral soil, and life above and below ground play such an essential role. So, and finally, integrating animals into land management. That could be grazing animals, birds, beneficial insects, and keystone species such as earthworms. So, and how do you think those various animals keep the soil healthy? Well, think of their excrement and the breakdown of the excrement, and the breakdown of plant matter through the hoofs, for instance, or as the animals eat it and trample that plant material into the soil, or their digging activities, nesting activities, and how they mix organic matter and aerate the soil. So why do these 5 soil health principles only seem to address soil biology? Well, I think it's mostly a result of the fact that we've forgotten or suppressed that the biosphere is so important in our soils, and that it's been replaced in agriculture with a lot of engineering. And secondly, soil biology is also the sphere that we can influence particularly well with reasonable success and in a short period of time. That's on the one hand why it's turned into an engineered environment in farms, but on the other hand, how we can turn that back and benefit now from much deeper biological knowledge to follow these 5 principles that lead to more biological ways that are in many environments more cost effective for soil health improvements. How do we use these 5 soil health principles? Well, one way is through monitoring. It's important because these principles lead us to indicators of soil health, both above ground and below ground. And monitoring can be done, for instance, through simple soil testing methods, and I'll discuss a few of them, and through monitoring of vegetation, for instance, through the Bullseye method developed by Kirk Godsey and Todd Graham in 2013, and which was adopted by my firm for woodland management in 2017. And this gives a very practical approach to how we can measure terrain and understand how the terrain speaks for the soil, you could say. And then the 5 soil health principles are, of course, important in applying management practices. So to give you some ideas of soil tests that we could do, you could actually identify the soil texture, so the amount of clay, silt, and sand, by wetting the soil and to then press it, roll it into a ball, and pressing that ball out with your thumb and forefinger to try to form a ribbon. If it forms a good ribbon, and it doesn't break until maybe after 2 inches, 5 centimeters, it has a high clay content, and then you're in the green part of this triangle, the soil texture triangle. If it makes a medium ribbon between 1 and 2 inches, it has more sand and more silt in it, and you're in that yellow-brownish component of the triangle. If it breaks before an inch, if you can make maybe a part that's half an inch or so, you're having a high sand content and a very low clay content. If you add a little bit more water to it, you can actually test for a grittiness. If it's very, very smooth, you have a lot of silt. If it's very gritty, you have a lot of sand. Neither gritty nor smooth, then you have clay. And that actually helps you also identify where you are in the subdivisions of these different colored parts of this triangle. So here, for instance, we did a soil test in that soil pit that I showed you earlier in the webinar. And based on the measured soil length, you can here see it's about 4 centimeters. We came out that this was a sandy clay. From nearby, I also took another sample, and we used a different approach in a settling column. You put it in a jar, shake it, and see how it settles out. And after 24 hours, you can see what you're having. The organic matter floats at the top of the water. The clay has been gradually deposited in that about centimeter coffee with milk colored component at the top. Then there is about 3 or 4 centimeters of silty material. And then at the bottom, 1 or 2 centimeters, we had sand. You cannot very much see that in this jar, but looking closer in reality, you could actually see the texture difference. And then analyzing that based on the triangle that I showed you, you come out to a silt loam. So the arroyo was slightly different than the soil under the pinon that was next to it. So in here, you can also do a soil acidity and alkalinity test. There are many different techniques. You can, for instance, buy a probe, but these probes are not very specific, although they show specificity, unless you buy one that is very expensive. And you still have to take soil samples from different depth, or from at least a rooting depth, but at least a dozen different locations, mix those all up, and then put your probe in it to actually have a representative testing. Another one that I really liked was shown in the following webinar, where you can at least identify, in the next video, where you can at least identify whether your soil is alkaline or acidic, and the level of how it fizzes identifies.
Half a cup of baking soda, which we're going to add here to see if it starts fizzing. We need to stir that a little bit and see what happens. This is the one with vinegar, and this is the one with baking soda. Well, if nothing happens, the soil is pretty much neutral. Which is a good sign, because that means that the soil has optimal plant growth capabilities.
So basically, the video wasn't ideal. Maybe it may not have come through right, but you can mix in one jar the soil with half a cup of vinegar, and in another jar the same type of soil with some water and half a cup of baking soda. And if they start to fizz, if one starts fizzing or the other, then you know what it is. If you have the soil with vinegar beginning to fizz, then it means you have alkaline soil, because the vinegar reacts with the alkalinity in soil, versus the baking soda would react with the acidity of the soil. And the amount of bubbles or reaction that you get says something about how alkaline or acidic the soil is, but that's only in a qualitative way, of course. Another method that I like very much was the cabbage method. You can find that online, where you actually use red cabbage water, and then you get a coloration of your soil in relation to the chemical reaction with the cabbage water. And that creates then a very pretty much distinct color pattern that, if you relate that with a color chart, you can actually identify how much the pH really is. And that is a pretty cheap and fun thing to do at home, and it's not as costly as buying all kind of probes or all kind of testing equipment. Okay, then a little bit about the Bullseye method. The Bullseye method was developed for rangeland health objectives, but can also be used for pinon juniper woodlands, especially, of course, when there is a significant grass component. Then you maybe don't even have to adapt it. But I have adapted it for forest health targets and tested it out in the last 5 years with clients such as Santa Fe County and here on the Glorieta Freedom Range, and it works really very well. It is a method to track change over many years. Monitoring is basically tracking progress and seeking early warning signals of desired or undesired changes. It's a pretty hands-on tool for those working directly with and on the land. So you don't need scientists really to help you interpret the data. It is a solution on the scale between no monitoring at all, if it's over your head to do that, or detailed technical qualitative monitoring. I mean, no monitoring at all, for instance, if you don't care to do it, and if the detailed technical quantitative monitoring is over your head. So therefore it's practical, but still science-based because all the indicators are based on research. It's qualitative and it's a rapid appraisal approach. And it leads to direct management recommendations. So it relates to the 5 soil health principles because you look at the above ground soil health, plant species vigor, plant health, and how the plants relate to the soil through a series of questions. You look at, for instance, the amount of litter on the ground. You look at plant spacing, etc. Soil health assessments are often quite technical and complicated to analyze. And this gives you pretty much through a comparison of good expressed in gold, which means achieving your goal, silver moving away or moving to your goal, or bronze not achieving your goal, 3 categories to figure out for each of the different indicators how you're actually doing. So, and that then gives you indicators for how the health of plants relates to and the health of the topsoil relates to the health of the deeper soil. So the full list of indicators is here along these spokes. You look at understory cover, the presence of topsoil, meaning the lack of erosion, the amount of litter, duff, dead and decadent herbaceous material and fine fuels, the amount of medium to coarse fuels, litter and fuels distribution, presence of soil life and soil moisture by digging into the soil a little bit and seeing how much moisture you have and how much life forms you can actually see. That by itself is already a pretty good qualitative indication of how much organic matter and biological activity conversions you have in the soil. You look, of course, at desired plant percentages, whether you have any non-native plants or not, or whether it's a monoculture. Tree species richness and functions, age diversity between the different tree species, tree health and vigor, are they sick, are they stressed, or very healthy? Ladder fuels and the crown height to get a sense of how flammable the landscape is, because that might over time impact your soil health, of course, because fire, especially if it gets too intense, will negatively impact your soil. Tree stem density, peak cover, understory vigor and function, the accessibility of the terrain, which is important to understand whether you can actually apply the management measures that are the result of this analysis, and the presence of living organisms on the soil. How much wildlife do you see, or how much small insects or worms or so do you really see in that area, because they all tell you something about the biological activity on and in the soil. And then you can feel and smell that soil, and under a microscope maybe see the mycorrhizal fungi, the hyphae, of the mycelium. When I mentioned earlier that maybe you can see them on the rootlets, then you don't see the individual hyphae of the fungi. You see actually clusters of them, and they make actually vibrate a little bit in the wind. So the clusters, the bundles are visible by eye, but the hyphae of the fungal threads, you could say, they are too small to see. You have to use a microscope. So that then eventually leads to identifying for each of those indicators how you're doing and the scoring of them on that bullseye. And you put a dot in the gold, the silver, or the bronze for each of those indicators. And when you've done that for all of them, you get a cluster and you see whether you're doing really well. Like in this example, we scored this pretty high, although for certain elements, you're not scoring so high. You're, for instance, in the gray or in the silver. That means that they need more attention, and you need to analyze and figure out how for those specific indicators, you may need to do some work. So here, for instance, in this landscape, we scored it pretty high and identified only that the amount of litter and duff and decadent herbaceous material was not ideal. The distribution was not ideal because it was often in piles. And the ladder fuels, there are ladder fuels in this landscape. So the problem of this particular part of the woodland is mostly that some organic matter is piled up and not really breaking down well enough. In other areas, it was not covering the soil ideally. And together, the entire structure made this landscape too flammable. So that then leads to management measures as, for instance, spreading out the organic matter, maybe thinning out the ladder fuels and distributing them over areas that are eroding. So that gives you an idea of how this bullseye method can be used to improving the landscape and has then also an effect on improving soil health. So that leads us to the end of this webinar. Thank you very much for being part of it. And we have time now for some questions.
Thank you, Jan Willem. Again, please take a moment to enter your questions into the chat box. I will go ahead and relay those questions to Jan Willem. And once he's kind of responded, then you can unmute and have a back and forth. He'll tell you to unmute. So if you have questions, now is the time. I've got my eyes on the chat. While people are kind of formulating questions, Jan Willem, I had a question. When you were talking about kind of building the water holding capacity of soil, you mentioned a list of things. The first one was adding not water, but organic material, which I think is going to help me remember that. But the next one after adding organic material was, try and minimize external inputs. So could you maybe talk a little bit about ways that you can add organic material to the soil, or that you recommend adding organic material to the soil without bringing in, being careful not to bring in external inputs to the soil?
Actually, the list was meant to show that you should not really start with water, although you need water, of course, for things to grow, but often just adding water to sedimentary soils, of which we have a lot here, on which we have difficulty to grow things, will not make much of a difference. And so a lot of soils first need organic matter, and then they will actually be able to retain that water or the compost tea or whatever you're adding to the soil. I think I was not clear in clarifying the external inputs, because that has nothing to do so much with this. or maybe I mentioned that the external inputs like a compost tea or so need to be added once you have some organic matter so that the compost tea can adhere to the organic matter mix, to the soil clots that have been forming between the mineral soil and the organic material. So once the organic material is mixed throughout the soil, you actually get a better structure with more pore openings in which the moisture can be retained in the soil. Because also the organic matter helps to form, of course, the sponge function that the soil eventually gets. And then it kickstarts biological activity with the microbes that live on the organic matter by nature of how it typically works with organic matter, especially if the organic matter is has been composting for a little bit or has been mixed with some manure or so, then you really kickstart life in the soil. Sometimes soils have to be inoculated so that you have to bring soil in, healthy topsoil from underneath the trees, where the microbiome, especially of fungi and bacteria, is present, and need to mix that with areas where it has been depleted as a result of erosion, for instance, or where you have gotten just pure sediment flow over the soil. And research is being done at UNM where actually they look at to what extent fungi or the soil microbiome stays intact even after trees like piñons or junipers have been removed by cutting or fire, and after how many years or decades or even centuries. And there is still, in many cases, there are fungi spores available, but you, like I said, you can kickstart that better by bringing that in those kind of depleted situations.
Very interesting. Thank you. Thank you for clarifying there. We have some questions now coming into the chat. So I'll start. Carl Struck was interesting, you know, could you talk a little bit about the ideal depth of chips on bare soil?
Yeah, it varies, of course, per site, but my experience with mulch in general in New Mexico on grasslands and piñon juniper soils is that it should not be too dense. If you actually have 1 to 2 inches of wood chips or wood mulch, then you're good. If you add more, then the plants will not regenerate through that very well. It will take many more years for plants to regenerate. But my rule of thumb is that you actually want to see a little bit of native soil through the mulch. And then you have an optimal situation for plants to regenerate. If actually your main goal is to initially make sure that there is no erosion or that you don't lose soil moisture through evaporation, then you can mulch a little thicker so that you maybe don't see any bare dirt beneath it. So also if the dirt is compacted, or if the topsoil is totally washed away, then you're actually in a situation where just the mulching by itself doesn't happen. You have to bring that organic matter into the soil. And of course you can do that by waiting, and it takes years for it to actually work itself into the soil through frost heave processes, for instance, and animal wildlife activities. But you can also manage it through maybe managed grazing, trample that area with livestock, or dig it under. For large landscapes, of course, that doesn't work. Then you just have to let it sit and wait 5 to 10 years until the organic matter is decomposed and that microbes and animals basically have brought it into the soil.
Thank you. I have another question here. Ryan Swazo Hines would like to know, how do you encourage vegetation growth in locations where thinning has occurred? for example, the removal of juniper?
Yeah, so in most native landscapes that are not overly disturbed, there is plenty of seed available in the topsoil. And it's a matter of waiting for the right conditions of temperature, daylight, and moisture for those seeds to germinate. And so what we've seen is that under certain canopy cover regimes, for instance, a canopy cover of pinon, juniper over 40 to 50% gives a dramatic drop in grass and herbaceous regeneration. So you have to thin a woodland pretty much to 40 to 50% canopy cover before you really have a good environment for herbaceous development. Your soil type, of course, will have to do with this too. In a coarser, sandier, or gravelier soil type, you have far less grass regeneration than on a finer, siltier, loamier soil. However, between rocks, for instance, on south-facing slopes, but then you have to have a pretty open canopy, grass tends to come up pretty well here around Santa Fe. So it is largely dependent on soil type and canopy cover opening. In areas that have largely depleted again its topsoil, you'll have to reseed the area, or if the area has been impacted by cheatgrass and other weeds, or has been burned, seeding with native grasses is very important so that you gradually start competing out the weeds, because the weeds will typically grow a short period of growing season, like cheatgrass is typically done in the first week of July. And then afterwards leaves pretty much an open bare area, especially if it's disturbed by wind or animals. So sowing with native grasses then is very important to make sure that you get a more permanent cover and stimulate the regeneration of more permanent plants that then also stimulate the microbiome underground for more permanent support of native herbaceous cover. That's a long answer, but I hope it helps.
Thank you. Next question here, Spencer Williams would like to know, what can be done to increase litter distribution?
Okay, so in pinon juniper woodlands, my experience is that if you, for instance, do thinning, it is really helpful not to remove the slash, but to spread the slash out in small, in branch size amounts, and basically do what is called a lop and scatter approach, where you drag the slash to the bare spot areas and cover those intercanopy openings, which are typically pretty bare, with the slash. That creates a microclimate. It gradually breaks down that slash material, and don't pile it high. It needs to just cover the soil so that you still see the bare soil between the slash. You may overlap it a little bit. It should not stick up more than a foot typically. And there are all kinds of techniques that you can orient it also to optimize erosion control. And that really helps. If there's old slash or fallen trees, spread it out. That typically helps. There's a lot of research that has been done over the last 20 years here in New Mexico that shows that the use of slash and how to spread the slash leads to microclimate improvements in the topsoil, helps with erosion control, and added organic matter and carbon components in the soil.
Thank you. We have a question here from Canyon Young. What would you say is the #1 management concern related to the pedosphere in PJ Woodlands?
The #1 management concern, I think there is not really number one. It depends so much, again, on the different types of pinon juniper ecologies. I think, for instance, in grassland and savannas, we've seen so much encroachment of pinon and juniper on those fine textured soils where actually we, by the nature of that landscape, would need to see more grass cover. That there, the problem is that we don't see enough diverse vegetation, leading then to a diverse and disturbed herbaceous cover, because we don't have as much grazing on those lands anymore, because the grazing industry here in New Mexico, for instance, has collapsed in the last 25 years. And so we have retained and suppressed so much fire in those grasslands and savannas that those natural ecologies have changed. So basically the litter layer and the A horizon have been depleted over time. Then in other woodlands, in some cases, we have seen too much erosion as a result of all kinds of disturbances, from use disturbances or upstream disturbances that caused erosion or flash flooding or increased flows. So sometimes you may see sediment packages, sometimes you see erosion. So drying out of the topsoil is then a major problem that leads then to decline of plant growth. I think it all comes down in many places to a decrease of maybe organic matter and a distribution of organic matter in healthy ways for the ecosystem. Maybe that is actually then the single most important one.
Awesome. Are there any other questions? Those are the questions that I've seen in the chat. Yes, we just had one come in. Carl Struck would like to know if you could discuss burning slash versus scattering slash and some of the pros and cons with those different methods.
Okay, so when you have a lot of slash, sometimes you have too much. And so when there is a lot, it is important to pile it and burn it. It is important to keep the pile small because the intensity of the fuel, and again, that is all dependent not only on the amount of fuel, but the size of the fuel, the size of the pile, temperature, soil moisture, and all that, may cause on that location of the burn pile, the pile to be too hot. And then you destroy basically the life in the topsoil and kill that area, sterilize it for a certain period of time. So actually, it has been shown in research that in a low intensity pile burn, the fungi will survive and after a couple of years kickstart the regeneration of plants on the burn pile area. So that should not at all be a concern if you do it right. Spreading the slash in some cases works and in some cases not, and it's very site specific depending on the landscape, moisture regimes, etc. But what is shown in general, it shows that lob and scatter works really fine. The newest research of the last 5 years also shows that mastication is a really good way of maintaining soil moisture by reduced evapotranspiration, or evaporation really, induced plant regeneration, minimizing invasive species, especially cheatgrass, and spreading that mulch, because depending on the type of masticator, you can actually have a rotary blade masticator that actually spreads the mulch in a radius of 30 feet around the equipment, which leads then to pretty good dispersal, which sometimes with slash management might be too costly or is not done well. So, but because again of the different soil types, the different types of pinon juniper woodland, all those different techniques have their variations and have different people have had different experiences with it in different locations. So it is important to look into it and then figure out for your specific location what would work best.
Thank you.
Any other question?
Yes, I have another question here. Miguel Licona would like to know, do pinon trees need to have soil on roots when relocating? And is there a time when they can be moved with bare roots? Must they be transplanted to equal quality soil?
With most perennial plants, it's important when you transplant them to actually move them with their root ball, because the soil associated with the roots has all this microbiome, all the organisms of the microbiome that are associated with that specific plant. And yes, piñons have a very specific set of fungi and microbes associated with their growth. So transplanting them with soil in the root ball is important. Second, transplanting plants, in my experience and from what I've read, is most important when the trees are dormant. And so that is best done over the winter between November and February. And of course, at a time that the soil is not frozen, and that you can dig it out without too much pain, because if the soil is frozen, you tend to just make the smallest root ball possible and cut a lot of roots and fungi out of the system that you transplant. And if you lose any soil material, try to replenish it with soils from where this pinon came from, so that it has its own microbiome that you carry with you and that you mix with those roots in the area where you plant that tree. That is all really important. Typically, and I've not much experience with it, but from a arborist point of view, you want to probably trim the tree a little bit, depending on the size, so that actually you don't stress it when it starts growing. And because the bigger the tree is, the greater your risk is that the tree will actually not survive in the transplant. And so trimming it back may actually help balance out its growth and metabolism with the root activity that is possible with the root ball that you're transplanting, because the root ball is of course impacted and has a lower capacity of supporting that tree after the transplant.
Thank you. I think those are all the questions that have come through at this point, Jan Willem. Would you like to take any others if folks are interested, or should we just?
Sure.
If anyone else has a final question, go ahead and enter it into the chat, please. If not, see, give folks.
Or if people have other experiences and want to share from that, I'm happy to hear that too. We're all here to learn, and I'm still learning a lot too. So.
See. Just give a minute or so to see if anyone's typing away on the other end.
So let's go to the last slide, because it has my email address. So that if people have questions, you can send it to JWJANSENS at ecotonelandscapeplanning.com. So JW Janssens at ecotonelandscapeplanning.com.
Okay. We have one question that just came in, again, kind of a final question here. Miguel Licona would like to know, how big of an area around a relocated tree from good soil to poorer soil must be prepped for the newcomer?
So typically with tree planting in New Mexico, we make the mistake that we try to make a deep, narrow hole. It is much better to make a wide hole, tree planting hole, so that actually the soil is loosened, but it needs to be deep enough that the entire root ball, without twisting the roots, can be put in the planting hole. So that also then offers the opportunity for optimal water infiltration in that planting hole. So ideally, the planting hole needs to be as large as the drip line of the tree, or maybe 1 or 2 feet wider than the drip line of the tree. That's a general tree planting rule in New Mexico, and I think that that definitely applies to piñons. So it depends a little bit on what soil you're planting that tree into. The greater the difference and distance from the origin of the tree to the tree planting hole, the more risk you run. And so the more you have to pamper that tree, or the more important it is to even have a relatively young tree that you're transplanting, like a seedling up to 4 or 5 feet tall. Because taller than that, you run more and more the risk that that tree needs more root support than you actually can transport over to the new hole. Other than that, I think if you plant it in a piñon juniper woodland, it will have, by its nature, a greater chance to probably establish, because of the soil ecosystem of the piñon juniper woodland that you're adding this piñon to. If it's outside a piñon juniper woodland area, you have even a harder time getting it well established, and it's even more important that you bring native soil over to support this tree and the microbiome that is in that soil to support the tree. I'm not quite sure whether it answers the question, but I'm willing to listen to a follow-on question or to entertain the question more by email.
Miguel says that yes, thank you. That's helpful. And then Carl just shared some experience that, you know, the addition of a 1/3 of an acre wildlife pond has made a big difference in biodiversity. That's in the chat there. I'm going to
I think that's absolutely correct. If you have a diverse landscape, including a riparian area, that will absolutely, for instance, attract birds and wildlife that then bring in an added biological activity onto the soil, into the woodland, or in a planting area.
Okay, I'm adding some information for keyword searching for the other webinars on YouTube into the chat. So for webinar 2, you can search measuring woodland soil health. And for webinar 3, you can search managing woodlands for soil health, and it'll be the top video that comes up. Oh, there we go. Nice. Thanks, Jan Willem. Just wanted to make sure that folks can find the others. Okay. Well, thank you so much, Jan Willem. It's always a pleasure, and I really enjoyed the second opportunity to see the webinar.
Well, thank you all for your participation, and thank you, Gabe, for doing the facilitation of it. And if people have any questions, feel free to email or call.
Awesome. Thank you. And with that, that is the end of the Healthy Soils Webinar 1. Thank you all for joining us. If you, like Jan Willem said, feel free to reach out with any questions.